Grüezi ragazzi, bienvenue und willkommen to Cannabis Scientia LinkedIn Live sessions. My name is Christina, I'm your moderator, and today I have the founder of Cannabis Scientia, Viola Brugnatelli, um, joining us today. We've had a few guest speakers, which were fantastic, but now we want to hear from you again. And today you'll be talking to us about mitochondria and how that fits into our endocannabinoid system. So I really look forward to our presentation. So without further ado, let's dive straight into it.
Thank you, Christina. Thanks again for lovely introduction, and thank you everyone that is watching live or rewatching this one today. Let me just add on. Thank you, thank you so much, and let's spoiler all of the presentation. Oh, there we go. So today, the concept and the idea behind, just as a little preface, I don't have a lot of time today. We are in a true live, so any question that is coming into your mind, please feel free to start placing it on the chat box. We're here to get your questions, and if I don't manage in the time that I have available today, we're anyway gonna collect them, and I'll if you write them there as a comment, I can go back and write you a reply, or you can stay up with Christina. So we already had a little intro together to just warm up and perhaps collected best your questions. Thank you. The idea and the concept behind today's class, which of course, as I said, it's a program that wants to be as a standalone class by class, but also as a sort of collective consciousness that together builds up one over the other, is the concept of how we learn on how to master our energy. Obviously, this can be observed from many points of view, and there are people with way much better tools in terms of, I don't know, Reiki masters and all sort of energy healers out there that can definitely give a better perspective than what I can share with you. But from my humble point of view, there will be a conduction through science. What is science in terms of the interconnection between the endocannabinoid system, mitochondria, which we slightly touched on already in the couple of previous classes with Dr. Moreno Sanz, and I will try to touch back again on this topic, and how it's linked back again to the concept of epigenetics. Epigenetics, sure, it's a quite a well-known term, but just as a reminder, etymology comes from Greek epigenetics, what's above genetics, so something that can change and shape the genetics. Usually in English, we talk of the nature versus nurture type of conversation. So um, obviously, as I said, my objective today is gonna try to explore this interconnection. And again, the scope for me is not just like to provide knowledge, which I think, again, I'm not, there are much better resources out there, but it's to help you think, to receive, material that can help you go and analyze perhaps certain aspects under different with different tools, and then to put them into practice, because there is a huge difference between talking about water and dipping your fingers in the fountain. So the hope for and the desire behind this program is to also take you by hand, and together try to build up a sustainable practice or different sustainable practice and open up to sustainable practice that can make us interact with biology in a way that is not just scholarly and is not just an overflow of information that goes at the intellectual level, but perhaps also that can enter in our bodies and in our energy fields. Obviously, again, as usual, if you want to dig deeper on the topic, especially of the endocannabinoid system, the book of reference that myself and Dr. Fabio have written and published is Principle of Clinical Cannabinology. Here is a QR for our digital book, but you can also get your fantastic, brilliant physical copy in your Amazon. So it really depends whether you're watching this from whatever state, then you're going to have to find this title in your .com, .da, .co, UK, depending from where your Amazon of reference is. So again, as usual, we don't want to bore you too much with the deep microbiology, so we're gonna keep it really light today. But so forgive me for like the super scientists for the hyper simplification of the explanation of mitochondria, but half an hour otherwise to try to put all of this concept together would be absolutely impossible. And for everyone, mitochondria's main function, you've maybe heard it already, it's that of allowing us to exchange what we give in terms of food, okay, and oxygen. So the main component that pretty much everyone understands that are necessary component for our survival, at least for our energetic survival. Into what's the token, the coin, that is used internally in our cellular system, so to create and generate cellular respirations. Here you have a view of mitochondria within a cell. So no, you can't see my cursor, but they are signaled by the is it literally written? It's the ones that look like bacteria, okay, and that are floating there in the cytoplasm. The reason I put this image is, at least for those that have never seen or a section of an animal cell, can start to get an idea of, where is the outside the membrane bilayer, now that envelops a cell, pretty much a main component of an animal cell, and then the distance of that to the most precious book that each of our cells is holding in the nuclei, and so where then part of our topic will take place, which is the level of genetics, and so where our book of genes is present, so our DNA. Now, talking of DNA and talking of genes, one of the unique features, and this was already mentioned a few times, but let's repeat it. One of the unique features of mitochondria is that they do have their own DNA, so their own genetic structure. We call it mtDNA. They also have their own set of ribosomes, simply meaning they have their own set of generator of proteins. But I think what's interesting in terms of this conversation today is that, first of all, mitochondria can replicate independently of the cell. So this is already a quite interesting and fascinating part, because it means that they don't necessarily depend from the health itself of the cell to support physiologically the cell, or as often we biologists, pharmacologists, doctors, whoever is working with usually pathologies, will look at mitochondria with systems that have distress. And so systems that usually are linking an issue with the capacity of the mitochondria to provide good cellular respiration, which is linked to a catastrophe of event, most often with the necrosis of the a malfunction of the cell, and then a necrosis of the cell. And often we see that related to pretty much an inflammatory pathology. And with inflammatory, I'm talking as well about neuroinflammation. We know there are links with Parkinson's disease, there are links with Alzheimer's disease, and so on. But obviously, today the focus is not about pathologies. This program is about physiology, how we're made, how potentially we can tap into how we can continue to preserve our physiology in the best possible way. So uh, the aim is how do we master these very important little organelles within ourselves. And also, and these are, please take it as a pure speculation driven from my own beliefs, um, how the fact that they are passed down maternally, and so they can in a sense be used for a sort of like ancestry tracing, because even if the mother, it's the biological mother of someone, it's unknown, we can find out much more about their genetics simply from our capacity to read how our mitochondrial DNA is written. So, in a sense, this time capsule not only encapsulates the energy capacity of our mothers, it encapsulates the energy capacity of their mothers, of their mothers, of their mothers. And so it's a powerful link, I believe, in my very speculative point of view, of the big love, maternal love, of that je ne sais quoi, that throughout any type of animal we see as instinctive maternal love that link and will link forever, even in type of animals or in type of human relationships, where the relationship between the biological mother and offspring are not necessarily colliding or collaborating that much, or being in a state of union, where quantically we are keeping this interplay and intercross of energies. And again, this is my speculation. This is one of the bases, I believe, on which why mothers can feel when something is going on with their kids, can feel when it's a moment to, and on this endocannabinoid system, again, we have quite a lot of literature showing the intercross and interplay between the fetus and the mother in terms of fetus development, after birth as well, on CB1 receptors present in the tongue of the offspring, and the 2-arachidonic glycerol present in the milk, for example, for mammals. So this constant interplay that there is between these two peculiar figures that are the offspring and the mother. And this is our gift, I think, from motherhood, from the mothers, that gives us the capacity to literally have energy in our body and to literally be doing anything we are doing. I'll try to speed up. Mitochondria don't just deal with respiration, cellular respiration. Again, if you're interested on this topic, I suggest to read a really good microbiology book. And get, I think this has been one of the first topics that have absolutely inspired me for mm, the incredible ways in which our cells have set us up, whether to aerobically or anaerobically, to allow us to exist. And the reason I say with in awe and just taken back from amazedness, because just like pretty much any other biological system, or protein kinases, uh, pathways, or whatever it's really happening within our bodies, has always shown me more and more and more the incredible refinement of of what we are built of, and how all of this, it's actually working, even when we have no idea how to define these things, even when we don't study and we don't know how to name any of these things, even when we had no tools to be watching mitochondria to understand how mitochondria worked. They were still already working for thousands of years and allowed life on this planet. So this is to say, what a gift. We are like a walking gift, and a walking incredible chemist. Now, the reason I wanted to put this image in here is because I think it's again, a hypersimplification of some of the main areas that mitochondria cover in terms of what they do. And for those that have more foundation on what also the endocannabinoid system does, you will immediately relate with this, know that these are some of the main key areas for the endocannabinoid system to be working at. If I go out, okay, like this is better, I don't cover the slide. Perfect. So obviously, last time again, Guillermo was citing the main Vincenzo Di Marzo definitions on sleep, feed, memory. Sorry, feed. Which one is the sleep, feed, memory, generation of new cells? I'm missing someone. A fifth one. Please write me in the comments the fifth one I'm missing, because today I haven't slept enough. But sorry, and organization of inflammation. I don't know if someone has read it, because I'm with the full slide. But exactly those that you see in the picture. So the areas on which mitochondria is working, calcium homeostasis. We know that the release of endocannabinoids is pretty much based on internal calcium release. And as of anything, and that's why we had a separate class actually talking on electrochemical balance and endocannabinoids, is because one of the ways to be looking at the endocannabinoid system is as a system that not only brings us at homeostasis, but also that has the capacity to shift what's our electrochemical frequency within the polarity of our own system, and without the magnetic fields of where we are operating. And so that's obviously done through the means of redox. Then other important actions that also coincide and are regulated by the endocannabinoid systems are the aging of a cell. We know it's fundamental for the separation and differentiation of stem cell production into, for example, glia or neurons, like giving indication of what this stem cell needs to become. That's already done at the fetus level. As I said, stem cell regulation also in the adult body, and then obviously this is super known and by any doctor that is successfully utilizing cannabinoids with different states of inflammation of their patients, whether they are neuroinflammations or bodily, like peripheric inflammations, or also like the main type of mm, pain statuses that we successfully utilize cannabinoids on tends to be obviously inflammatory pain over, let's say, acute pain. ATP production, ATP I just gave for granted that is a known word, but it goes for adenosine triphosphate. Could be any word. I think it's a crypto. It's the crypto. It's the internal token that our cells are using, and it defines energy. It's like it's the token that it's needed by our cells. Sorry, I move my hands because ultimately I'm clearly very Italian. Okay. Thank you, Mariana. Yes. The relaxed part. It's probably because I haven't relaxed that much and I was forgetting that part. Thank you so much. Thank you so much to the community. Really, it's a common effort today. Yeah. Again, the energy output is estimated around 36 ATP molecules from one molecule of glucose. This is not a perfect equation. Obviously, in biology, nothing is. But I think it's particularly relevant. And the reason that I wanted to put it out here today is because, again, this is something that can be changed, because we can have less productive mitochondria, usually in a pathologic state, or in a state in which we don't see pathology, but the mitochondria is underworking. And so usually we see a pathology when all the symptoms are exploded, but before all the symptoms are exploded, a lot of things have been happening for quite a lot of time in terms of deceleration of our system, in terms of unmastering of our energy. So it's almost certain that before a certain group, let's take Parkinson's, for example. Before we notice symptoms, by the time we notice symptoms, there is already like an important cell death that have been gone around. But for that group of cells to be unresponsive or not being able to produce enough dopamine that we know is needed and is part of the hallmarks of the pathology, months or even years before, the certain type of activities have been going around, obviously not just in the mitochondria, but mitochondria are highly involved, because without energy, that's pretty much the no prana, no game. Okay, borrowing a word from Ayurvedic medicine. Again, the heat part, it's for anyone that remember that also our cannabinoid system deals and has in the cannabinoidome the vanilloid receptors. So also those receptors that are ion gated channel receptors, and that deal obviously with hydrogen, so with acidity, and therefore with inflammation, internal states of inflammation, but also with our internal thermostat of internal thermostat and also external thermostat. No, so the TRPV1 and TRPV3, especially when we touch like a surface that is too hot, like we are going to need to receive like really fast actions and fast messages of, hey, take your hand off the hub. That's not really a good idea. You're gonna damage the tissue. So we know that the endocannabinoidome, the endocannabinoid system, it's also controlling and supporting the production and the maintenance of homeostasis through so many different levels, and also heat. It's one of those, and mitochondria are quite fundamental in these two. Now, let's get on to, I'm really running out of time. Let's get on to one of the papers that have been cited the other time by Guillermo. This is from the group of Giovanni Marsicano, as he was mentioning, because it is indeed the main group that has been efficiently and working on this such interesting topic of the cannabinoid receptor present at mitochondria. The reason why I wanted to retouch on this topic is that, of course, the other time I think Christina was asking on whether there were structural differences. There are, and in fact, genetically they are defined as slightly different. MTCB1, there is not much fantasy in the naming of receptors in this case, as much as in the naming of medicines. But it's believed that they are involved with the modulation not only of energy production, but also being energy production in all the system of synaptic, and going back to synaptic transmission. Then we can relate back with the topic of memory. If you remember, when we started to talk about how we need certain, different type of action potential for one cell to communicate to another cell, with to communicate to another cell, and then a group of cells, then an orchestra of cells to start to communicate coherently into what's the creation of a new signal. And so the shift of what we called short-term plasticity into a long-term plasticity. So the shift from something that actually takes quite a lot of effort, these action potential that need to oscillate and change vibrationally and electrochemically, the next one, and then the next one, and this action, we need to trigger it and then be able to sustain it enough and repeat this cycle enough times so that it moves into a way less effort requiring, way less glucose required to enter then long-term plasticity. So to enter a state of something that we have learned. This is commonly explained in anyone that wants to approach more the study of how the hippocampus, and so the main area dedicated to learning and memory, works. And so it's clear that when we look at the mitochondria present in those type of areas, and the cannabinoid receptors, that are flourishing in there, the connection is not so speculative at that point, that they are going to have quite an important impact in energy use. But then, as we're building also all of these classes, how energy and memory and cognition, they are not so abstract to our, again, emotional balance, because we have already layered up knowledge from the first classes that really were about the intercross and interplay between the prefrontal cortex and the amygdala, and so emotional balance as well. It's a slightly more recent paper, where simply I can make a easier, and less speculative conversation on how we can induce changes in the respiration of brain mitochondria, and then how these changes can be brought about by whether external cannabinoid stimulation, but also internal cannabinoid stimulation, so endocannabinoid stimulation, because already the fact that epigenetics is the capacity of our body to turn genes on and off with the different energy status. Realistically, we know already how, for example, endocannabinoids or external cannabinoids have quite straightforward or direct or a bit indirect ways to be talking to our genes. This can be done, as I said, more indirectly. So through the main, cannabinoid receptors that are present in the membrane, external membrane bilayer, lipid layer of the cell, and then from there, the G protein cascades down, protein kinases cascades that in turn, at the end of all of this, will be talking to the nuclei. So we can expect changes that perhaps are going to be a bit more ahead, later in time. Then we have changes that are a bit more direct that the cannabinoid receptors do in terms of their stimulation and their direct activation or inhibition of certain genes. And this typically, the example that usually I bring about with this is the PPR receptors. So the group of, again, cannabinoid receptors in the so-called endocannabinoidome, no, the big group when we look in large at the endocannabinoid system of receptors that are present directly in the membrane of the nuclei and that act, in a sense, as transcription factors. So as indicators for the certain genes present in the nuclei to switch on, to activate, to transform into proteins, or to shut up, to not be transforming that gene into, translate, actually, that would be the word, into a protein. And again, a lot of literature can already exemplify as different type of diets and stress and toxin and stressors go and alter gene expression. We could list so many different diseases that are affected by epigenetics. So the changes that externally we are receiving, stress, chronicized, for example, and how we can transform these into pretty much any pathology that you can think of, chronic pathology that you can think of, from cancers to neurodegenerative disease. But again, today I wanted to focus on stuff that I think it's more relatable for anyone present here, which not necessarily is going through pathology, but might be going through fatigue, brain fog, neurodegeneration in terms of like spiking too much glutamate, and therefore not really doing the best favor to their own cognition at that time. And hey, it happens to everyone. And the point is, how do I stay in touch enough with my system, and I receive messages, and I put myself in a receival capacity to listen to the messages, if you want, of my mitochondria, and how do I work back this energy, and so how do I master back this energy so that I can avoid fatigue, brain fog, and therefore then something more dangerous like potentially neurodegeneration. I wanted to add this quote: mitochondria don't just make energy, they help decide which of your genes are active, and that's epigenetics in action. On this concept, as much as we know that external circumstances can influence our biology, and that's what we call epigenetics. So we can generate the external circumstance. We can literally be the external circumstance for our system. And this is what I am trying to arrive to the last slide so that we can do this little exercise together to be the external circumstance that goes and change the mitochondria. I will touch on this quickly. It's very speculative, but some type of literature start to point at quantum physics for the capacity of basically electrons moving in mitochondria, because it's really what's happening with the generation of ATP, uh, to exist simultaneously in multiple states, and to maintain synchronized phases, and that this energy transfer, it's way too efficient to be happening without this happening in what now biology calls quantum coherence. So an oscillation um, that that is improved and protected by what usually doesn't really allow for such functional um, energy circulation, because it tends to be like a more stagnant environment, like the wetness and warmth of the inner side of a cell. But it is thought, and there are some papers that are looking at that the inner lining of the mitochondria perhaps is where an energy, the main manner in which electrons are moving around and particles are moving around, and how it's optimized this process is through electrons oscillations, and this allows more um, rhythmic membrane potential. What I want to say with this, stay with me, please stay with me. I just look again. This doesn't want to be like the science, okay? This wants to be some science that I bring here to you, and then your brain can potentially make connections, or perhaps have, you know, a nice dinner conversation at your dinner table, or it just doesn't go anywhere and just leave it there. It's speculation of different things that, in my head at least, they make sense. So we know that other systems are working through these oscillations, okay? We know that the endocannabinoid system modulates mitochondrial function. We have started to see this. We have evidences of this. We know that CB1 receptors on mitochondria can influence directly the output of ATP and the balance of ATP, or of reduction oxidation, the redox balance. And so, in a sense, we can say the endocannabinoid system could potentially stabilize this such quantum coherence via those membrane oscillations. Potentially, that's the place. Maybe it's all happening on a matter-wise of empty CB1, but also this is, you know, how it's happening as such a quick, optimized way. Okay, and again, it is very possible that in systems that require groups of cells to communicate quite efficiently, like for example throughout neuroplasticity, that this is one of the methods that is utilized for this interplay, and so that again, in the role of the ECS of maintaining homeostasis, there is a capacity of the endocannabinoid system of being present at the so that put probably the fact that right now there is mainly one group in the entire world that is looking and observing and researching on mitochondrial cannabinoid receptors is, I believe, an issue that we all have, because we should gather way more knowledge on this, because I think they actually have the key, they're withholding the key for much more knowledge that we can think of. Now, as I wrote here, speculative but intriguing. Um, it is possible that for those that want to explore the electromagnetic field of consciousness, the so-called semi theory field, um, this is one of the ways through which not only, as we said already, the endocannabinoid system shifts, as we said, our electrochemical balance at large groups of neurons that are firing together, let's say communication between the amygdala to the prefrontal and then back from prefrontal to amygdala or to the nucleus accumbens or badi, but also at the level of single cells. And so at the level of the single cells, what do we have inside? Well, those cells need to work. They need energy. And so the energy output of the single cell also is very important within the system. And other systems that are working and are using this capacity to interplay with the oscillatory fields, we know them already, and they are the navigation of birds with the magnetoreception, photosynthesis as well. There is quite coherent energy transfer. And another very fascinating topic for a neuroscientist is the whole topic, huge, we leave this aside, of the vibrational tunneling detection that is done through, I believe what's, if I'm not wrong, it's the Mayer group of receptors present for one of our sensory senses, for one of our senses, which actually is olfaction. So olfaction is the sense for which we have been presented with the majority of neurons. And again, but if you're listening to me through headphones, and in this moment I am generating oscillation and vibration, pretty much everything, no, the screen that you're watching, that all of this is working through that way. So it's not that far-fetched to be imagining this as well, in the direction of microbiology and how energy systems are working. To end our session today, and I think we put enough for at least some nice dinner conversations, I wanted to introduce you to a very basic pranayama technique. It's a pranayama. It's just breath work. I borrow it from Ayurveda, which I'm knowledge about. I've been working in a Ayurvedic hospital in my life, and I think on this then we will be able to build up eventually other breathing techniques like Kapalabhati, like breath of fire, that will give you even bigger and immediate indication of how we can shift energy and respiratory capacity of our cells in a quite sudden way. But because they are kind of advanced exercise, I wanted to start with something that will benefit any of us, whatever state we're at. It's called Nadi Shodhana, or alternate nostril breathing. Here I wrote, it calms the mind, balances the autonomic nervous system, it improves oxygenation and cellular energy. And again, even if nothing is believed out of this class because it was way too speculative for your ears, at least you can come out of it with a little breathing exercise that in any case will benefit you for the next hours. So even if you don't believe that through this exercise we are actually changing the biologies of our body, and so therefore that we are in charge of constituting and creating that environment that then signals and does the work, the epigenetics that signal to our systems, just enjoy this practice. Okay, so I think as usual perhaps I deactivate my no I'll stay on camera so you can have a reference point on what I do with my nose. I'll try to conduce you on these. As usual, if you want to, I don't know, have a stand up and uh, just remember that you have a body and that you have a spine, and perhaps your spine was from many hours like kind of rounded, and you don't even remember anymore that your neck is like this. Like this is a good moment for you to uncross your legs and get your neck straight so that your end of your spine and your belly button and your heart space and your neck and your head are aligned so that we basically have a little bit more lung capacity. And then today we just do a very simple with our hand. Look at me. I'm just gonna use the in English, does it have a name? No, maybe it's just called fourth finger. Sorry, I don't know what language I'm speaking today. I had meetings in different languages, so I assume this is called fourth or ring finger. I don't know. And it's beyond me. But this finger, but yeah, I think it's called the ring finger. This one on the left one, and obviously, if you're left-handed, do it with your left hand, okay? Do it how you're comfortable. I'm gonna be using my right hand because I'm right-handed. And so I go with the right thumb on my right nostril and with my ring finger on my left nostril. And what I will be doing is literally alternating the closing and the opening of the nostrils, so that if you remember the type of breathing that we have started to teach throughout these classes, have been breathing where we inhale, we hold, and then we exhale, and then we hold, and then again. Okay, and so the pattern is the same. We're going to be inhaling from the right side, we're going to be holding, we are going to be exhaling. Okay, and simply we're going to be doing this with the use of our hand. Today we're not going to be doing weird stuff with the hands. This is not needed. It's just for you to get the basic of this technique and to try to remember to keep your head straight and to just start to enter, start to dim a bit your eyes, and perhaps also close your eyes. I will try to conduce you. So bring your hand close to your nostrils, and then close your left nostril, left side, ring finger, inhale with the right side. We inhale for 4, we close both of them for 4, and then we exhale 4, 3, 2, 1 on the left side, and then again we hold. Try to straighten up your body, and then we inhale 4, 3, 2, 1, and then we hold, and then we exhale. We hold, we inhale 4, 3, 2, 1. We hold, we exhale. 2, 1. We hold, we exhale. We inhale, sorry. We hold, we exhale. We hold, we inhale again, right side. We hold. We exhale again. We hold. One thing that we tend to do, keep on going. One thing we tend to do is that with the elbow, we tend to just fall with our body. Try to remember that and keep your back straight as much as you can. Again, inhale, right side. Hold. Exhale, left side. Hold. Inhale again. Last couple of cycles. Bring it up. Hold. Exhale, left side. Hold. Last cycle. Inhale. Hold. Exhale. Left side. Close and hold. And then gently, you can take your hand off, but try to keep your eyes still closed, your back still straight. Try to feel a bit how is your body now? Where is that? Whether something has shifted a bit. And just for you to know, something I wanted to leave you with in terms of a little quote, that inside your cells, each and every one of your cells, your mitochondria might not just be chemical factories. They might be humming with quantum music. Humming. It's a lovely onomatopoeic word. A word that if you think and you have some different spiritual or religion connections, you've been hearing around a lot in Sanskrit, in Latin, amen. It's the word of what many define God. Whether your God it's Gaia, then that's up to you. So if your God is biology, that's also great. But yeah, thank you for joining me in this closure on how we can move and shift energy moving in sync, how we can resync when we feel out of sync, the tools that we have at hand for that, on perhaps refacilitating ourselves to bring in current energy, cellular respiration, and from there systems coherence. Sorry, before we completely get off camera, I wanted, how do I present? Thank you. I just wanted to say goodbye with my usual back, just reminding you as well that you can also write me directly at viola@cannabiscienza.com, especially because I went so out of world that, yes, I literally have a couple of minutes and then I need to shoot out. So very happy to take any question, or otherwise I'll leave you to the incredible hands of Christina. Thank you for intervening and taking me out of my madness. So
Another fascinating talk. It's too bad you don't have time for questions. I already have a question, but I guess I'll email it to you after. But yeah,
In
mainly,
the comments, whatever that is,
yeah,
because
yeah, in the comments for sure. So just like, um, just thinking of the mitochondria and the electron transport chain, I think if I remember correctly, there's also an uncoupling protein. So, and that kind of changes, well, it changes everything because it changes the flow of electrons. So, just as a speculation from you, how do you think that would affect um, the quantum biology of our body? How does that change what we signal out?
I guess we need to have a long speculation dinner on this, because also I don't know if we already posted this or not. No, perhaps after this live is gonna go live, but also on histones and their movement there as well. There could be like an interesting trigger on the differences of how these oscillations work. Again, this is extremely speculatory. I think another part that made me observe all of this under this lens is, again, if you have the patience of looking how this mitochondrial DNA is shaped. If you see it's
Mm.
shaped a bit differently than the usual genetic
Nuclear.
material that we were used to see, unless we take in, and obviously this is considered quite from quite a lot of people as the reality of the science. The theory that there's been like bacterial co-engineering with our body, because then obviously it looks way more like potentially genetic material that we would see in that type of organisms than in ours. Um, Again, I think shapes are not that unimportant when we think of function. So no, and we know this in biology, and then again, function that defines as well um, movements, that defines oscillations, that defines, and that's why I was going into the direction of now of the humming, because then that's at least perceivable v- differences in vibration. I have never read, like going back to your questions, I've never necessarily read anything that explores like that specific topic related to this topic on the, the quantum biology that I wanted to start touching on with today's topic. But perhaps there is some literature in that direction, and I'm keen to check it out. And if I do find it, I'll make sure to follow up with some links. Yeah.
And maybe I can do the research too, and I'll let you know. How
For
about that? We'll
everyone,
come up
because I believe
with
this is
some
a very
homework. Yeah.
collective, yeah, it's I think it's a very collective space. Like the other times also came out some questions that made me actually the questions that you've done to Dr. Moreno Sanz made me then feel inspired onto the building up of this one. So it's again the collective that is cooperating and working together. So we're a good orchestra, just like ourselves.
Yes, collaboration is the way to go. It's what our body does. It's what we do as humans. We tend to think of ourselves as individuals, separate, but I think one of the themes emerging from this course, and also just my general exploration of meditation in life right now, is that we are all like, small versions, and then I guess it's called fractals. Am I going like the small pattern and then bigger, and it's still the bigger pattern. So mitochondria, cells, system, body, and then beyond our body, like we are also part of the system of a community. So that's why collaboration is definitely the way to go. I'm not a fan of competition. It's not really, I believe that we can achieve everything better through collaboration than competition. but to each their own. And just want to say a shout out to Mariana. We
THC,
actually,
THC. Recently, I was in a conference in Capitol Hill, and the theme was we all need a bit more THC in our systems: transparency, harmonization, and collaboration. So I fully agree on that, and perhaps someone could also say it is potentially this separation is a potential illusion as well, because yes, we can look at the breakdown of small to large and macro to micro, but then again, going back on Julie's talk, uh, that in itself it's a dualism that not necessarily it's a correspondence of what is very philosophical, but as well could be very scientific. Um, yeah, sorry.
philosophy is at the root of all science because it teaches us to question and explore. So I really personally, I really enjoyed that you bring these concepts together in this course. I think that, and that's one of the reasons why I'm so excited to be part of this. It's really, it's unique. It's unique. There are not many people, especially like as scientists, once we train as scientists, and if we end up working in the scientific field for a long time, we kind of get stuck in this reductionist approach where
Kant
we take,
Kantian approach, as in Kant,
and
like
yes, yes,
okay. Yes,
yeah, like
where it's a very masculine energy, I would say, of like labeling and dividing, because it's great as a great use when we group, then we can work with those systems, right? But then we also need to, I believe, have the capacity, just like the endocannabinoid system, to bring together and to collaborate and make those different groups communicate one to each other. And I think in this, the ECS is a great connector, because it truly connects different systems that don't look like they could communicate one to each other, yet they do. So, and I think this is the part that fascinates the most the doctors that we train, because at least for how it is taught medicine in Italy, but I believe this is widespread in many parts of the world, it tends to be very compartmentalized. So if I'm an eye doctor, then I just look at eyes, and if I'm a neurologist, and each their own, and that's great because then we can get highly specialized. But then the moment that they start to study the physiology of the endocannabinoid system, some of them go in kind of crisis. They're like, but why are we looking at this part, and how is that connecting? I think it's much easier from the eyes of a biologist, perhaps, to be having this as a standard concept of, yeah, when you break it down, at the end, the cell is a cell. And if you break it even further, then you're going to have smaller particles, and then smaller particles, and then you arrive to waves and blah, blah, blah. Sorry.
and I think the endocannabinoid dome, it teaches us to shift our perspective away from the reductionist towards a more integrative approach. Because when we try to study the endocannabinoid system with our reductionist scientific tools, we keep getting snubbed. And it's like, yes, but they're also connected here, and the connection here just kind of shifts everything. I often think about it as like a chain mail, like or like little bowls sitting on water, but they're all connected with chains. So when you pull one, it changes, shifts everything, but at the end of the day, they're all still trying to stay floating on the surface. So yeah, that's kind of,
I like the
that's one
metaphor.
thing. Yeah. Are there any other questions? Because we're having a private conversation here, which is wonderful. But I want to give our audience members the chance to ask any questions. And as I mentioned before, nothing comes to mind now. Just comment on this thread, on this video, and Viola will come back and answer all your questions. And then there was also a question here by Simone. He asked, Can I see it again? Is there a registration? So yes, all of our sessions are recorded. You will be able to find the recordings either on the Cannabis Yedzia LinkedIn page and also on YouTube. I pasted the link there. And don't forget, there's another session next week. Yes, Viola, go ahead.
No, sorry. I announcement.
Yes.
Next week, I want to take a well-deserved spring break or Eastern break. So there won't be a session next week. Too much travel. I just no. I need a little spring break, but we will be back in not next Tuesday, but the one later with other guests, and again, it's gonna be a duo, so just keeping you entertained. There will be again a duo of scientists that will bring about an interplay of pragmatism, um, practical tools on how to work on our biology, and also coming with some hard science. Like we are trying not to stray away too much from that, by still giving pragmatic tools. So next Tuesday there won't be another live, but in 2 Tuesday there will be, and we're gonna be back full force.
So our next session is going to be on April 22nd. But don't worry, if you get
Yes.
the invites from me, I always invite everyone like one by one, so you will not miss it. I will send you invites. But yeah, thank you everyone so much for attending today. Thanks, Viola, for yet another stimulating food for thought session. And we'll see you all on the 22nd. Have a great day,
Thank you
everyone.
so much, Christina. Have a good day too.